today is Bioenergetics, and the topic of discussion is ATP, which stands for adenosine triphosphate. Objective for today is that students will be able to comprehend about the structure and function of main energy source of cells. First of all, we'll go towards a recap of the previous lesson. The first question of our recap is define redox reactions. The second question is explain stable and unstable associations. We have already gone through this. I hope you might be very clear with these terms. So the answers are redox reactions are those which actually involve exchange of electrons between atoms. And you know there are two types. The loss of electron is called oxidation, while the gain of electron is called reduction. Then the next question answer, stable association. Those made with oxygen atoms are not good source of energy. And unstable association are those which are made with other atoms. For example, carbon and hydrogen. They make unstable associations, and when they try to move back to oxygen, energy is released. I hope these terms are clear to all of you now. Now today, the major bullets of what we're going to discuss is about the major energy currency of cells. Major energy of cell is nucleotide called as ATP, which is adenosine triphosphate. We'll be discussing about cellular functions, synthesis of macromolecules like DNA, RNA, and proteins. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. Transmission of nerve impulses, active transport, exocytosis, and endocytosis. We are going to take a terminologies recap. First of all, let's talk about nucleotides. They form the basic structural unit of nucleic acids, such as DNA. They are molecules consisting of nucleoside and phosphate groups. Now, to clear the term nucleotide and nucleoside, have a look at this figure. As you can see, a nucleoside is basically a combination of sugar and a base. Nucleotide is a combination of sugar, base, as well as phosphorus. Moving on to nucleic acid, you can see there is a chain consisting of phosphate, sugar, base, and again phosphate, sugar, base, and this makes long linear chains. So nucleoside, nucleotide, and nucleic acid. I hope this is clear. Next term is cellular functions. Now, what is cellular function? Which actually means cellular respiration, which is a set of metabolic reactions and processes that take place in the cells of organisms to convert chemical energy from nutrients into ATP. As you can see, in mitochondria, cellular respiration takes place, and this is how our cells actually gets energy to do different functions. Then there was transmission of nerve impulses, which basically means the conduction of messages taking place through the neuron. And you know that neuron is the basic unit of the brain, and you are familiar with this figure as well. This is the structure of a neuron. Then let's discuss about active transport. It is basically the movement of the molecules from an area of lower concentration to the area of higher concentration. In the figure, you can see the molecules moving from lower concentration to the area of higher concentration. Next, exocytosis and endocytosis. Exocytosis is a process in which the bulky material is exported outside. You can see in the figure as well. And endocytosis is the process of cellular ingestion of bulky materials by the infolding of the cell membrane. You can also clearly see how it is getting inside, penetrating into the cell. Let's talk about the history of ATP. ATP was discovered in 1929 by Carl Lohmann and was supposed to be the main energy transfer molecule in cells. It was followed by the Nobel Prize winner Fritz Lipman in 1941. We have the picture of Carl Lohmann and Fritz Lipman. Let's talk about the structure of ATP. As you can see, the structure of ATP is based on three basic structures. I'll discuss about the first structure, that is adenosine. Now, adenosine is basically a nitrogenous base, which is actually showing you two rings. I'll highlight that area to make sure that we are talking about adenosine in this figure. This is adenosine. Now, moving on to the second structure, that is ribose. Ribose is basically a 5-carbon sugar, as you can see in the picture. It's highlighted. And this is basically a sugar molecule. Next, moving towards the third structure. As you can see, these are the high energy bonds, and there are actually 3 phosphate molecules attached. So these are the 3 structures of ATP, adenine, ribose, and 3 phosphate groups. Now let's talk about TILD. TILD, as you can see the symbol, is a connecting covalent bond which actually connects the 3 molecules of the phosphate. I'll show you how it is connected. As you can see, every phosphate molecule is connected by a TILD, which is a covalent bond connecting 2 phosphates, and the figure clearly shows the symbol. Now, let's talk about reactions. The first reaction shows you that ATP is sized up by a water molecule. When it is broken down, on the other side, the product that we get is ADP, which shows a removal of molecule of phosphate. And every removal of phosphate molecule is actually releasing 7.3 kilocalories of energy per mole. Similarly, when ADP is actually utilized in the second phase, we can see that adding up with the water molecules, it is also releasing 1 molecule of the phosphate, and it is reduced as adenosine monophosphate. And it again releases an energy of 7.3 kilocalories per mole. Next, as you can see in this figure, a structure of adenosine triphosphate is shown to you. Now you can see the figure over here, which is highlighting that the battery is completely charged. So ATP triphosphate is the best source of energy and the ready source of energy with lots of energy. And when it is reduced to adenosine diphosphate, you can see that due to losing 1 molecule of phosphate, it is converted to diphosphate, and there is a release of free energy in the picture as well, which is in the form of 7.3 kilocalories per energy. And this shows that the energy consumption is now decreasing. Now let's focus on this ATP cycle. As you can see, adenosine triphosphate, when it loses a molecule of phosphate in the form of energy, we release 7.3 kilocalories of energy, and it is reduced as adenosine diphosphate. And when adenosine diphosphate is added up with a phosphate molecule and the energy, it again regenerates and turns itself into adenosine triphosphate. So this is basically the cyclic process. And that the adenosine diphosphate can be easily converted into adenosine triphosphate. So the recap of today's topic is major energy currency of cells that we have discussed. We have discussed about the 3 basic parts, that is adenine, ribose sugar, and phosphate groups. We also know that 7.3 kilocalories of energy is released every time a cell breaks. And you know that ATP formation is a cyclic process which takes place by the cyclic of ATP to ADP, and then again we can regenerate ATP. Well, there's a small activity for all of you. I want all of you to draw the structure of cells energy currency on your notebooks as a practice so that you are more easily going to understand the concept. So this is the end of the lesson for today. Thank you very much for listening, and I hope that you might have grasped the concept of ATP.